expectations. Sorry. So welcome back to botany class. Okay, botany second year class. So we are discussing about the SAQs now. Okay, already we have finished the LAQs from the 13th chapter and the 14th chapter. We are going to have the SAQs right now. So in the SAQs, the bacteria chapter was finished. There is a 4 different SAQs are there in that chapter. What we have finished, there is the shapes of the bacteria and the arrangement of the flagellum on the bacteria and the nutrition in the bacteria, and the last one is the conjugation process in the bacteria. What we have finished. Now we will start with the next chapter, that is the virus. So in the virus, the expected question, first one is the structure of TMV. What we have to concentrate here. So how to write the answer and how to draw the diagram. Both are important here. What we will lose the marks if we won't write in a proper way. Everything is having the systematic way. You have to follow this, just like the traffic rules for us. So in this answer, what we have to write? What is TMV first of all? It's a tobacco mosaic virus. TMV is a tobacco mosaic virus. Tobacco mosaic virus. So what we have to start like this. TMV means it's a tobacco mosaic virus which causes infections in the tobacco plant. Then the structure of, directly now we can start the structure of TMV. Okay, so in drawing the structure, so what it is having? It is having a central core first. You can draw this oval shaped structure. Just surrounding to this, the capsomers are present. We can draw like this very easily now. Okay, maintain the line over here. Like this only, it should not go like broad. Now, wherever you have missed, you can draw the capsomers. That's it. Now, we are drawing with the pencil, so you can erase this line also, whatever you have drawn as a outline for this. Okay, it should not have the very much broad, and the diagram should be finished within 2 seconds when we are drawing. Make sure that don't waste much more time over here only. Okay, and now we have to draw the genetic material that is present in this. Easy one. We don't want just like a print from the reader, because I'm saying for the supply students right now, not for the complete academic year now. So in this, I can expect what is your drawing capacities and everything. Based on that only I'm saying. Okay, so you can draw like this simple, or else if you don't know how to draw this also, don't bother. just to draw a spring-like structure from the top. That is also enough. No need of any artistic one for this. I don't want. No one want. Whoever are correcting. Okay. So this is the main structure. Now, what we have to say here. So the viral organisms, basically what we know, viral organism is a non-living organism having the protein coat for it, protein which are making the covering the viral organism. So if we know this, then we can easily say the points in the TMV. So the very first point is the TMV means tobacco mosaic virus, which causes infection for the tobacco plants. Next sentence, there is a second point what we have to write. The length of this virus is 300 nanometers. Length, I'm saying the length. Okay, length of this virus is 300 nanometers. Width of the virus is 18 to 19 nanometers. And this much only we have to write in the second point. Now comes to the next point. The virus is having a central opening called as core. Central core. Okay. So in that same point, we have to write the diameter of the central core is 4 nanometers. This is the diameter of the central core. That is 4 nanometers. The diameter of the central core is 4 nanometers. Now that point finished. Okay. Now we have to know what are these, what we have drawn. These are called as the capsomers. So the complete body is covered with the protein subunits called as capsomers. So each and how many capsomers are present in this TMV? Capsomers are of 2130 in number. Capsomers are 2130 in number. Okay, how many capsomers? The entire body is covered with the protein subunits called as capsomers, and the capsomers are of 2130 in number. Each and every capsomer is having 158 amino acids in them. Every capsomer is having 158 amino acids in them. Okay. For each and every amino acid is capsomer, sorry. I'm saying something. Every capsomer is having 158 amino acids in them. So like that, how many capsomers are there? 2130. Okay. Those many amino acids. No need of the further thing. Now we have to write which type of genetic material is present in the organism. In the central core, which is having a opening like structure for the virus, for the TMV. In this central core, the complete genetic material is present in the spring-like manner. The genetic material in the TMV is single-stranded RNA. The genetic material in the TMV is single-stranded RNA. And here, when we are writing the single-stranded, the two SS will be the small case. It should not be the capital letters. Single stranded. Okay. This represents, one is the single, second one is the stranded. No need to write the complete word also. Small s RNA. And when you are writing the RNA, make sure these three letters will be the capital letters. If in this any one letter is the small letter, then the complete word meaning changes and you won't get the full marks. Here, if this one, if you have written like this, then we can easily deduct 1 mark for this letter itself. That much important. And if you ask us also, we can say very easily because it is a genetic material and what you have written is an enzyme. Completely different. And we have deducted the marks. So don't go with that arguments or any other thing. So whatever we are saying, listen properly and implement this properly. That's it. No further arguments on this case. Okay. So always the genetic material should be written with the capital letters. All three letters should be the capital letters. If any one letter goes off, that means if it is the small letter, then it represents the enzyme. It's not the genetic material. So the genetic material in the TMV is a single-stranded RNA. Okay. And in this RNA, how many nucleotides are there? Complete genetic material is having 6,500 nucleotides in them. This much if you have written over. So how to write this now? In a systematic manner. Okay. So TMV, structure of TMV. The question is structure of TMV. TMV means it's a tobacco mosaic virus which causes infection to the tobacco plant. So this organism, this viral organism is having the body of the length 300 nanometers and the width of 18 to 19 nanometers and having a central core inside them that is having the 4 nanometers in diameter. Okay, the central core diameter is 4 nanometers and the body is completely covered with the protein subunits called as the capsomers, and the capsomers are of 2130 in number. And the molecular weight of this is the TMV is 39 into 10 to the power of 6 Daltons. After the capsomer number, what we have to write? The molecular weight of the TMV is 39 into 10 to the power of 6 Daltons, and each and every capsomer is having 158 amino acids in them, and the genetic material in the TMV is single-stranded RNA, which is present in a spring-like manner in the central core. This is single-stranded RNA, which is having 6,500 nucleotides in it. That's it. We can wind up the answer. That means 5 to 6 points we have to write and one diagram with the labeling done 4 out of 4. Because supply paper will be somewhat, they will made it as somewhat easy to clear by every student. Okay. Those who has having the backlogs, at least one paper, only one paper, then they can easily clear and they can join with the other students for the higher studies at the same time. So no further lapse of the academic year. That's our intention of the board. So for that, we can expect the easiest questions only. Okay. Sometimes we'll have the hard questions, but if you select the chapters and in that chapter, every question, if you prepare, then you can easily go for the IP exam, for the supply exam, where you can score nearly 45 marks very easily. Only 45, that is a first class marks. From the failure to the first class, it's a very nice thing for us. Okay. And now comes the second one in the viral organisms. That is the bacteriophage. Structure of bacteriophage. So what is meant by bacteriophage? Bacteriophage is a viral organism which is infecting the bacteria. That is called as the bacteriophage. It is having the tadpole shape. The shape of this is tadpole shape. Okay. Now comes the diagram. First of all, I will draw the diagram, and in simple way I am drawing so that every student can draw and they can remember the parts what we have to explain in the theory, because in the material they have not given the explanation for the diagram, but here we have to give the explanation for the diagram itself. See, just like the hexagonal shape, we have to draw first and double line for the hexagonal from the inside and a small cone-like shape at this point, that is just like a collar for the shirts. Okay. So the name also collar only, and the tail part and the spider legs like structures we have to draw. So what are they? I will say right now. Okay, this is the diagram for the bacteriophage. So the bacteriophage is having a tadpole shape, and this body of this bacteriophage can be divided into two parts, head and tail portion. Head and tail portion. Okay, so this head and tail we have to write on the left side of the diagram. Right side you have to give the proper labeling. Both should not be combined. And the labeling part is, this is the capsid, and in the center of the capsid it is having the double stranded DNA, and this is collar, this is core, and this red color one is the tail plate, and these are the tail pins, and these structures are the tail fibers. Tail fibers. Okay. So here the entire viral organism bacteriophage divided into two parts, head and the tail portion. Head is having it is made up of a thick protein coat called as capsid, which is having the genetic material inside this. The genetic material is the double-stranded DNA, and the tail portion is having the core and the tail plate, tail pins, and the tail fibers. And in between the tail and the head, a small collar-like structure is present, which is separating these two parts in the bacteriophage, and the functions of the collar are not yet known. Okay, and it helps out, mostly it helps out in holding the head outside the host cell, so that it will be present like this, the head, and the other part of the virus is present inside the host to make the separation, to hold the head at the top. It will help it out what we are knowing till now, but the functions of the collar yet to be known till today. And what we are saying that we are too clever and we made everything clear. It's not like that. The virus is too clever than us. It will changes its shape. What we are facing since 3 years, Corona, same candidate. Okay, so we cannot say the original shape and everything, characteristic features, the same thing for this also. And the tail plate is having the tail pins and the tail fibers. Tail pins are total 6 tail pins are present. Okay, here you can see only 4. The other one will be behind this because this is the LS of the bacteriophage. That is the longitudinal section of the viral organism. So the other one pin of the both the sides will for both the legs are present at the back of it. So here we can see only 4, but actually those are of 6 tail pins and 6 tail fibers are present. And the tail fibers main function is to hold the host cell, and the tail pin main function is to pierce the cell membrane, otherwise the cell wall of the host cell, and makes it a free entry into the host cell. And the head, that is having the capsid. Capsid main function is to release the enzymes into the host so that it will the cell membrane will disintegrate and the viral organism can enter easily, and it can easily pass the genetic material of the virus into the host cell to cause the infections. This is the main function of the head and the tail part. So what we have to draw, what we have to say is very clear. Bacteriophage. Bacteriophage is having the tadpole-like structure, and this organism will infect the bacteria. So we have given the name for this viral organism as the bacteriophage. And this bacteriophage body can be divided into 2 major parts, head and the tail part. Head is hexagonal in shape, having a thick capsid and the double-stranded DNA in it. And the tail portion is having the core, tail plate, tail pins, and the tail fibers. And the head and the tail portion are separated with the collar-like structure. And the core is the empty space, which is having a hollow space in between them, so that it can easily make the passage of the genetic material into the host cell. And the tail fibers and the tail pins are meant to hold the host cell to give the infection by this virus. This much you have to write for this bacteriophage. Very easiest one, what we can say in the botany itself. And based on this 2 questions only, one, the third question, what now we are going to discuss, that is the chemical nature of virus. Chemical nature means how the body is made up of. That's the one. So this in the material, that means in the list of the questions, this will be the second question. But I have said the TMB structure and the bacteriophage so that it will be very easy for you to understand. If you have not prepared this, also you can able to give the answer. How you can able to give? If you remember the basic nature of the capsid and the core in the two answers, you can write this answer. So I am saying this at the after that answers only. So the chemical nature, virus is made up of core and capsid. If you have seen in the bacteriophage and the TMV, we are having the central core and this is the capsid. And here also capsid and the core. That's the one what we are going to say in the chemical nature of the viral organisms. Viral organisms, chemical nature, it is made up of core and the capsid. What is meant by core and what is meant by capsid? Okay, so the core is the central part, that is a hollow tube-like structure that is present in the viral organism, and where it is holding its, otherwise it's making the passage of the genetic material from the viral organism into the host cell. Okay, and this core and the capsid both are made up of the protein coat, and this protein coat is the proteins can make themselves as the enzymes. Otherwise, they are the another form of proteins is the enzymes. So they can easily release the enzymes now. So that's the way how they will infect to the other host organisms. And next comes the capsid. Capsid is made up of the protein coat, which is very hard and gives the specific shape for the viral organism. And the capsid is the one which is holding the main genetic material in it. So the genetic material of the viral organism may be the DNA or RNA. DNA or RNA. So in this DNA also, DNA may be the double-stranded DNA or single-stranded DNA. Single-stranded DNA. And RNA is of double-stranded RNA or single-stranded RNA. Any one type of genetic material only present in the viral organism. That may be the DNA or the RNA. If the DNA is present, DNA may be the double-stranded or single-stranded. If RNA present, RNA might be the double-stranded, otherwise the single-stranded. Mostly the viral organisms having the genetic material based on their host cells. To which organism they will cause the infections. Mostly based on that, their genetic material is going to be seen. Very rarely they will be having the other than the host cell genetic material. Example, HIV. Okay, it is causing the infections to the human beings. Human beings means we are having the genetic material as double-stranded DNA, but the HIV virus is having the genetic material as double-stranded RNA, which is a very dangerous one. So it leads to the death of the organisms only. And another example where what we are saying, the host genetic material and the viral organism, that is a causative organism genetic material is same. So in that case, we have to say it as a poliovirus. Poliovirus is having the single-stranded RNA and that is a double-stranded DNA, and the host is also having the double-stranded DNA. Like that. Otherwise, we can say the very best example, TMV, tobacco mosaic virus. Okay, which is having the single-stranded RNA, and it is giving the infection to the tobacco plant, where this plant is also having the single-stranded RNA. So like that also we can give the example. So this is the third question what we can say. Now comes with the one more question in the viral organisms. That is the lytic cycle of the life cycle of viral organism. So in the question may be the explain the lytic cycle of the viral organism. Okay. Otherwise, they can ask us the complete life cycle of the viral organism. Any one question they can ask. In this, we cannot guess. Mostly, this type of question we won't get in the supply exams. Maximum. That is, there is no chance of getting like this question 90%. 10% I cannot say. So that 10% I cannot take any chance right now. So I'm explaining this. So those who have not yet prepared, just if you know it, if you have attempted, otherwise written some points, what you heard right now, that is also sufficient to get at least 1.5 mark for 4 marks. 1.5 mark means at least we will make it as 2. Sometimes these 2 marks are worthful for us. We don't know what happens in the next moment, isn't it? So not to lose the marks, so we have to prepare with the almost all answers from the specific chapters where we are preparing to get nearly 35 to 40 marks. That is considered as a passing mark only, because botany is the easiest one. Okay. So lytic cycle. So in the lytic cycle means the life cycle of the viral organism, which occurs this lytic cycle, otherwise the life cycle, we can say for the bacteriophage. That is the bacteria, the viral organism which infects the bacteria. Okay. Inside the bacteria, the life cycle of this one occurs. So here it is having the 5 different steps. The first one is the attachment. Attachment. Attachment means when the viral organism attaches itself, otherwise attacks the bacteria and holds the bacteria, is called as the attachment. This is the bacteria, which is a rod-shaped bacteria, that is a bacilli type. And for this, the viral organism gives the infection. How? First of all, it attaches to the bacteria like this. And this bacteria is having a mucilaginous sheath externally. So I am drawing with the orange color the mucilaginous sheath so that the cell wall can be very clear what I have drawn with the white color. It will be very clearly visible to you right now, isn't it? This is the first step, attachment. When the viral organism infects, otherwise attaches itself to the bacteria with its tail fibers, is called as the attachment. And during this process, the tail fibers will release some enzymes on the mucilaginous sheath of the bacteria. mucilaginous sheath of the bacteria, and they will bend a little bit to hold the bacteria with its tail fibers. When they are inserting themselves, then they are stretched. Once they goes into the mucilaginous layer, immediately they will hold it, they will turn and hold the bacteria like this. That is the attachment to the bacteria. Now comes the second step, is the penetration. Penetration. Penetration means what we know. They will be injecting themselves. They will make they will pierce the cell wall. They will make the hole and enters into it. That is called as the penetration. So how to say this here? When the bacteria is like this, having the genophore and the plasmid and the cell wall and the plasma membrane for it. Okay, and having the mucilaginous layer also. Now, this virus, see how I have drawn previous, now how I am drawing it. So in the penetration, what happens? The tail pins which are present for the bacteriophage. These tail pins and the tail plate will releases the enzymes on the bacterial cell wall in a much more concentrated way, so that the cell wall will disintegrate at that place and makes so that the tail pins can go into the cell wall through the plasma membrane into the cell, into the cytoplasm, so that the tail plate also goes. So that means completely tail plate, tail pin goes into the bacterial body. So this will be done by the release of the enzymes over here. So which type of enzyme they will release? Lysozyme. They will release lysozyme enzyme. Okay, so that it's very easy for the viral organism to enter into the bacterial cell. Okay, so through the penetration process. So what happens in the penetration process? The lysozyme enzyme will be released in a large quantity from the cell plate, from the tail plate and the tail pins on the cell wall and the plasma membrane, and the both will disintegrate, and it makes the easy entry of the tail portion in the cytoplasm of the bacteria. So that is called as the penetration. Now comes the third step. What is the third step? That is the maturation. Maturation means once the tail part enters into the bacteria, immediately the genetic material from the head enters into the bacteria. The genetic material of the viral organism now passes from here and enters into the cytoplasm of the bacteria. Now what happens? The bacteria which is present like this, having its own genophore and the plasmid DNA, and having the mucilaginous layer. Now, this viral organism the viral DNA enters through the tail into the bacteria. Now what happens? Immediately the viral DNA makes the bacterial DNA into the disintegration. That means it removes, it takes out the completely, removes the bacterial DNA, including all the cell organelles of the bacteria in the cytoplasm. That means the bacterial DNA will not be there and the plasmid will not be there. So it is having only the cytoplasm, and the viral DNA. Now the viral DNA will makes itself as a fragments in the cytoplasm of the bacteria. Immediately the cytoplasm of the bacteria will think that that's its genetic material which is free. So each and every specific amount of the genetic material is surrounded with the specific amount of the cytoplasm. Once it is surrounded by the cytoplasm, it will takes up the energy from the cytoplasm and starts making the viral heads in this. So with this, what happens now? Viral heads are going to be formed. And somewhere else we'll see the genetic material like this. This is the one. And once this is completely, the complete genetic material enters into the bacteria, then the viral organism is the empty viral organism is going to be seen. Empty viral organism means it does not have any other genetic material. So the empty viral organism is called as the ghost. And the viral DNA that is present inside the cytoplasm starts getting the energy from the cytoplasm of the bacteria and starts making the new viral organisms. New viral organisms. Okay, this is the maturation. Now comes the fourth step, that is a biosynthesis. What is meant by this? Bio means life. Life of the viral organism starts. So at this step, what happens? The bacteria enlarges by the presence of many viral organisms in them. Sometimes the virus is having the tail also. Sometimes only viral heads are present in a large number. So like this, now there is a life for the virus. Usually what we'll call viral organism is a non-living entity. But here we are saying the side heading only as the biosynthesis. Means what we are saying, life is going to be synthesized. Where is the life? Here we are saying the viral organisms are the non-living entities. Here once the host is living, till that viral organism is having a life. Once the host cell dies, then there is no life for the viral organism. So the life of the viral organism starts here by making the many number of the viral heads with the tails, otherwise only the viral heads, so that the bacterial size increases. If you remember the size of the bacteria, what I have drawn in the previous, there is a very thin sized bacteria. Now it's a bulge because it is having many number of the viral heads in it. So it will become bulge. So once the synthesis of the viral heads occurs in the bacteria, immediately these viral heads are having the protein coat called as capsid now. That will releases the enzyme lysozyme. Lysozyme enzyme was released by these viral heads on the cell wall of the bacteria. So what happens with this? With this, at one place, the bacterial cell wall will burst. Okay, that is the last step. Last step is the lysis. Lysis means death of the cell. Now, once the lysozyme enzyme was more in the number on the cell wall of the bacteria, at one place, bacterial cell wall gets disintegrated and releasing the viral organisms into the host cell. This entire life cycle occurs in the major host cell. There is a primary host. Bacteria is a secondary host. In that, viral organism is living, just like we. In our body, bacterial are there. And to that bacteria, viral gets infection. With that, we are having the typhoid fever. So this entire life cycle represents the, for us only, the typhoid fever. So the typhoid fever duration is of 2 weeks, that is 15 days. So what happens in this 15 days? Every fever can be controlled. Malaria can be controlled within 4 or 5 days, maximum 5, minimum 4 days. Course is antibiotic course is of 5 days. But for the viral fever, antibiotic course is only 10 days. Why? Because here we have to kill 2 organisms, bacteria and the virus. So that is the one what we are saying over here. Okay. So the lysozyme enzyme was released by the viral heads. With that, bacterial cell wall disintegrates and releases the viral heads in the host cell, which so that these viral heads are ready to infect another bacteria and they can easily give the infection to the primary host. That is the lysis. That is the lysogenic lytic cycle of the viral organism. Okay. If you have written these 5 steps and drawn these 5 diagrams and say these are the viral head and these are the bacteriophage. And here, lysis of cell wall of bacteria. Done. Over. That's it. If you have written this, if you know the process, then we can easily give the answer for these questions. So first of all, logical thinking is mandatory. Okay. If traffic jam is there, then what we will do? We will search, we will see from here only. If the traffic lights are there and the traffic jam was occurred over there, then here only we'll stand on the 2-wheeler and we'll have a look and we will try to take a turn here itself. We don't want to go there now. Same logic you have to apply here. How to get good marks and how to remember each and every step. So the first one in the bacterial life cycle, the bacteriophage life cycle is the attachment, holding. Next, penetration goes into the bacteria and maturation. That means making the viral heads. Fourth one, is the biosynthesis. That means the viral heads were fully formed and the viral bacteria is completely filled up with the viral heads in them. And the last one is the lysis, death of the bacteria. With that, we can easily finish off with this answer and you can score 4 out of 4 very easily. There is no chance of missing off 1 point or half mark also in this. Okay. So with this, we have finished the 2 chapters SAQs. That means 1 SAQ and 1 VSAQ. 1 VSAQ, which type of VSAQs will get? That is what is the genetic material of the bacteriophage and what is the shape of the bacteriophage. Shape of the bacteriophage is the tadpole. Genetic material is double-stranded DNA. What is the shape and the genetic material of the TMV? TMV shape is the rod-shaped viral organism and the genetic material is single-stranded RNA. Like this only. What is the ghost? Ghost is the empty viral organism which is present on the bacteria during the lytic cycle. That's it. These 3 questions only we can expect from the virus and the other 3 questions from the bacteria. That is what is plasmid and what is its significance. Plasmid means a extra genetic material, circular double-stranded extra genetic material that is present in the bacteria is called as the plasmid. And this plasmid is what is the significance? It is useful in the rDNA technology. What we know, same thing if you have written, done. So like that, the VSAQs are there. Just go through it. And if you have any such thing, that means any such problem, you can give the comments in the comment box. Otherwise, we'll discuss the VSAQs all together in the at the last. That means once we have finished off the 5 SAQs, what you have to write. Okay, so 1 SAQ was finished. Now we'll go with the second SAQ, that is the genetics chapter from tomorrow onwards. Okay, right. Thank you. Stay tuned to this same channel, same time, so that we can clear the second SAQ also. Thank you, students.